So this is the beginning of Chapter 6's lecture on the cell. And this chapter is focusing on the structure and functions of the individual organelles and parts of a cell. But before we can look at that, we need to step back and see how do scientists study the cell, and that's through the use and with the help of microscopes. So you need to have a cursory understanding of the basic types of microscopes that are being used out there. And basically, what you should know is the one that we commonly use in the classroom is just a light microscope in which we use visible light pass through a specimen and then look at it through a glass lens. And the three parameters of microscopy, no matter what we're using to look at things, are magnification, resolution, and contrast. And we adjust these things. With magnification, it's the ratio of the object's image size to its real size. So we can change that by looking through different lenses. The resolution is the measure of the clarity of the image, how clear. So we adjust that with the fine tuning and try and get as clear an image as possible. And then contrast is the visible difference in brightness between parts of the sample. And we can adjust that by adjusting the light source and making it dimmer or brighter. So this is just a comparison of the different ways that we can view something. You can see here that light microscopes magnify effectively to about 1,000 times the size of the actual thing that you're looking at. And the resolution of a standard light microscope is obviously going to be too low to study organelles. So the membrane-enclosed structures in the eukaryotic cell is going to need something like electron microscopy in order to view those. So this helps you get an idea of the use of different magnification tools. So to get down to that smaller level to look at organelles, we have two basic types of electron microscopes, and we abbreviate that with an EM. We have a scanning electron microscope, or an SEM, which focuses a beam of electrons onto the surface of what you're looking at, and it gives you an image that looks 3D, and that's the one here on the right. There's also a TEM, which is a transmission electron microscope, and that focuses a beam of electrons through the specimen. And actually, this is what's used more to study the internal structure of a cell, and it's a larger piece of machinery like you see here. So now, what are we looking at with those microscopes? Hopefully, what we're looking at is the different types of cells and what they're made up of. So as a review, we have two types of eukaryotic cells. So we can have a prokaryotic cell also. But for most of this lecture and most of your Chapter 6, information is going to focus on eukaryotic cells. So eukaryotic cells are broken into either animal or plant cells, and prokaryotic cells are primarily going to be bacteria cells. And the big difference between prokaryotic and eukaryotic cells is the presence of organelles. Do take time to look at specific differences between prokaryotic and eukaryotic cells. Make yourself a little chart to make sure that you know what do prokaryotes have in common with eukaryotes, and there's a much longer list of what they do not have. So just so you can get that started, what they do have in common are chromosomes. So both prokaryotes and eukaryotes have chromosomes. So they have some sort of DNA. They both have ribosomes. They both have a plasma membrane, and they both have cytosol, or what we sometimes refer to as cytoplasm. But for all of the different types of cells, a more general term to use would be cytosol. So the only organisms, again, that are going to be Prokaryotic cells are going to be in the domains of bacteria or archaea. And then eukaryotic cells are going to include the domains or the groupings of organisms that are protists, fungi, animals, and plants. And you should also know that plants and prokaryotic cells also have one more thing in common. Plant cells all have a cell wall, and so do bacteria cells. So we have organelles in eukaryotic cells, which is a distinguishing feature that they have from prokaryotes. And those different organelles have internal membranes that actually surround them, and in that way make compartments or compartmentalize the function of the different organelles. Some basic features again. This is listing what I was just saying before, the things that are common between eukaryotic and prokaryotic cells. So again, make sure you know this list. But your Chapter 6.2 is going into just focus on eukaryotic cells and the organelles that are surrounded by plasma membranes. So before we look at that, here's a close-up picture of prokaryotic cells. Remember that they do not have a nucleus. Instead, their DNA is found in a region called a nucleoid, and that word is right here. So that's where you would find the DNA of a prokaryotic cell. They do not have membrane-bound organelles. Instead, their cytoplasm or cytosol is bounded by the plasma membrane that surrounds it, and then outside of that is a cell wall. Some bacteria or prokaryotes have flagella, but not all, so don't make that mistake to think that all of them do. Okay, so now eukaryotic cells, what they're characterized with having is DNA that is bound in a nucleus, and that nucleus has a membranous nuclear envelope or a nuclear membrane. The organelles in a eukaryotic cell are membrane bound, and the cytoplasm is in the region between the plasma membrane and the nucleus. Another distinguishing feature is that eukaryotic cells are generally much larger than prokaryotic cells. And in the case of a eukaryotic cell, the plasma membrane is a selective barrier that allows passage of oxygen, nutrients, and waste to provide for the entire volume of the cell. So this is a picture diagram showing a eukaryotic cell with its internal membrane and that is partitioning the cell into its different organelles. And this is a good picture showing the different organelles that you should be familiar with. There are many types of specialized cells in eukaryotic cells. So this one here is a human cell from the lining of a uterus. And you can see here the nucleus is very clearly stained. And you can see the nuclear membrane that's surrounding it. And then you can also even see the nucleolus, which is located within the nucleus. And you can't really make out the organelles very well with this magnification, but you do get a sense of the outer cell membrane. And you can see here there are cilia in this type of cell attached here. And then you can get a sense that there's cytoplasm in there. This is a picture of a plant cell, which is also eukaryotic. And again, there will be a lot of similar organelles to an animal cell, but there are some differences, and you can see some of those differences are peroxisomes, chloroplasts, and a very large central vacuole. So those would be the major differences that you would find between plant and animal cells. And of course, the cell wall outside of the cell membrane. So this is a picture showing you cells from duckweed. So obviously, this would be a plant cell. And this one is using an electron microscope. So you can start to see some organelles here. So you can see the cell wall is outlined in black. And all of these structures here are all the chloroplasts. You can see the orange structures are the mitochondria. The nucleus is the larger violet-colored circle, and the nucleolus is the dark purple there. And an important thing to notice with this picture is that there are a number of organelles of the same type in each cell. And that's an important feature of learning about the makeup of eukaryotic and prokaryotic cells. In eukaryotic cells, we have a lot of specialization. So that means the cell is going to develop and have the organelles needed to carry out its particular function. So, for example, with this chloroplast assembly that you see here, all those different chloroplasts tell you that this plant cell is going to be in a leaf. You're not going to find this plant cell in the root of the plant because there wouldn't be a need for photosynthesis in the root, only in the leaf structure. And so in this case, you would find in a leaf structure a lot of chloroplasts if you were to count all these. And you also have the presence of a number of mitochondria. So back when we first learn about cells when we're little, that we learn, there's a mitochondria, there's a chloroplast, there is the nucleus. So we learn a very simplistic model where there's one of everything. But in reality, it's much different than that. So while we're thinking about plant cells, this is a good time to talk about the cell walls of plants. So that cell wall is outside. It's a structure that is surrounding the cell membrane, and it's an important part of plant cells. It distinguishes them from animal cells, but it is something that has in common with prokaryotes. Also, fungi have a cell wall, and there are some unicellular eukaryotes that have cell walls, but most animal cells do not. So the cell wall is there to protect the plant cell. It's really important in maintaining the shape of the plant cell, and it also is very important in preventing excessive um, uptake of water or the swelling of a plant cell by taking in too much water. And when we start to look more into osmosis and diffusion, this will become very clear why this is such an important factor. And plant cell walls are composed of cellulose fibers. So we've been learning, and you've been building your cell membrane models. And the cell membrane, as you guys know very well, is made up of lipids, and specifically phospholipids. But a cell wall is actually made up of cellulose fibers, which, as you know, are carbohydrates. So it is a very different structure than your cell membrane. This picture shows you a yeast cell, and this is a eukaryotic cell that does have a cell wall. So I wanted to point out that there are some simple eukaryotes that do have a cell wall as well as the cell membrane. And it's interesting to point out that a yeast also has a large vacuole. So it does have something in common with a plant cell, a eukaryotic plant cell. But yeast are actually neither animals nor plants. They actually are their own kingdom.